last but not least talk of session 4, Mark will present the relation between reproductive development and physiological measures of condition, results of a laboratory experiment with black sea bass.
Okay, thank you. I guess I'm used to going last with a last name W-U, so I won't take it personally. First, I'd like to acknowledge my co-authors here, Rich McBride, who works with me in Woods Hole, and also Gary Fitzhugh at the Panama City Lab in Florida. So today I'm going to talk about some reproductive relations between reproductive development and with some physiological measures of condition, and sort of describe a small, very simple lab experiment we had done to look at this. This species was introduced yesterday by Nikolai, so it may seem familiar. So why we were interested in fish condition effects on reproductive potential has been well discussed at this meeting. There are often seasonal time lags in condition and in relation to the reproductive seasonality. And the nice diagram that Cindy Van Dam presented yesterday shows that to some degree. And as we've seen in this talk just preceding me by Lindsay, we can estimate some of these things at the population level, but we also, sometimes we need a lot of samples, and we need to sometimes look at monthly samples to determine the appropriate metric and time to look at, and we may need many years of contrast to look at good condition and poor condition and relate those to years of good recruitment and poor recruitment. When we consider estimating it at the individual level, in the field we're limited to one point in time per individual, so that limits the analyses to a large extent. And given the lags that are present in allocation strategies, the factors that we're interested in can often be out of phase and thus appear not correlated. In the laboratory, we can have multiple measures per individual. However, these are usually limited to simple measures such as length, weight, where we can calculate relative condition, and we can track those over time. There's the more informative measures we're often interested in, like lipid and energy content. Those tend to be lethal measures. But recently some new non-lethal technologies have been proposed to provide information on the energetic status of fish. And Lindsay showed a picture of the fat meter, which is one. And another technology that I'm going to talk about here is the bioelectrical impedance analysis. And this has been applied to human condition for decades. It is a relatively new development as applied to fishes. Cox and Hartman came out with a paper recently and then followed up with this. And one of the measures we can get from this bioelectrical impedance analysis is a phase angle, and that has been proposed as an index of metabolic condition based upon the reactance and resistance. And what this diagram shows here is we have two sets of electrodes. They're signal and detector electrodes, and these a small current is passed through, and it's measuring the reactance, which is proportional to the extracellular fluid, and the resistance, which is proportional to some of the cell integrity and cell wall structures that resist the flow of electricity. So we have been interested in this, and we're able to tag along with some, or include this in a study that was looking at other aspects of tilefish in the south, and this was in the Gulf of Mexico. And what we saw, seasonal patterns in this BIA phase angle that related to the spawning seasonality. So our observation that we saw this pattern, and we rose the question, is the individual variability in phase angle leading up to the spawning season related to their reproductive output, and how might we be able to use this as an indicator of reproductive potential? So, as the ongoing quest for proxies to estimate reproductive potential, you know, as I explained earlier, we explored this in this simple, we explored the utility of both simple and more detailed measures of reproductive output in a laboratory experiment. We specifically tested this bioelectrical impedance analysis, and our goal was to predict reproductive potential of an individual at time t from earlier measures of physiological condition at earlier times. So and then we were able to, at the end, sort of compare this approach to predicting reproductive output from both simple and detailed measures taken only at the terminal measure. So as Nikolai indicated yesterday, black sea bass, it's a continental shelf species along the east coast of North America, and it's managed in 3 stocks, which I've got highlighted here in the circles. Nikolai was talking about this Southern Atlantic stock. I'm going to focus today on this North Atlantic stock, although we did also receive fish from the Gulf of Mexico that were included in the laboratory experiment. I'm not going to talk about those fish today, although there was some mention of them in the abstracts. In this North Atlantic population, there is an onshore, offshore, and north-south movements. They move offshore in winter when temperatures get cold, so they are much more migratory than both the southern stocks. So we're also interested in how energetic allocation plays into that. A little more life history. This is a protogynous hermaphrodite. Sexual transition is believed to occur after the spawning season, well in advance of the next spawning season. They have asynchronous oocyte development and indeterminate fecundity. As some of the talks have been mentioned yesterday, Costa and also Cindy, we begin to question in the north whether they may be determinate, but we'll have more information on that moving forward. They're a clear batch spawner, multiple batches, some estimates of over 20 batches per season. So it makes it difficult to estimate the egg production by any individual. Just some additional differences between the stocks. I won't go into this in too much detail, but they're not terribly long-lived. They mature relatively early. Within a couple of years, they begin to transition at relatively small sizes. And the main difference here is the spawning seasonality. In the South and Gulf, it's very long, whereas in the North Atlantic, the northern stock that I'm focusing on has a much restricted spawning season, June to August. So just a little bit about our experiment. These fish were collected near Woods Hole, Massachusetts, where these experiments were carried out, and they were collected in the fall before out-migration, before they left the area. And this would be several months after the spawning season and give us time to rear them before the next spawning season. And I'm just going to discuss data for these 38 individuals that were from the northern population. Given potential sex change that's been demonstrated for the species when they're held without males, we divided fish into populations that had large, presumably male individuals. And we held them under natural photoperiod and a temperate water supply to try to mimic the conditions they would experience. And the goal of this rather simple experiment was just to get a range in condition and see how that related to our measurement of using this new technology. So we just tried to induce this by separating fish with 2 different feeding rations. We had a high ration where we fed them to satiation 3 times a week, and this was done with herring and gel food, and a low ration group that got 2 times per week that was squid and gel food. And we used the gel food, so it was a nutritionally balanced diet. We were not really trying to alter the food content. Because of the fish are pretty aggressive, we couldn't do fixed rations. We had to feed to satiation and control the number of feedings. And the individual fish, we anesthetized them, tagged them, and measured them monthly for length, weight, and this bioelectrical impedance analysis. And just a demonstration of how we do this. This is the fish board we developed. We spent the first year or more of looking at multiple fishes. We've developed this measuring board to help stabilize the readings. It reduces a lot of the measurement error in how we use this technology. And it's been purported to be pretty simple to use, and I guess you can say it's so simple a caveman can use it. And this is just a picture of some of the fish in the tanks we've shown. So at the end of this experiment, after 6 months, we sacrificed the fish, and in addition to those measures we had been obtaining all along of length, weight, and the bioelectrical impedance analysis, we got terminal measures of sex, the maturity class, the gonad histology, gonad mass, liver mass, viscera mass, and I have an asterisk here because we weren't able to get that for all individuals, but as was shown, we included this viscera mass because they have quite a bit of mesenteric fat, as was seen in the just previous talk, and we wanted to try to quantify that, but since we didn't have it for all individuals, I didn't include it in what I'm presenting today. It's a quite nice measure sometimes for laboratory fish where we're not feeding them in the days before we measure it, so their stomachs are and intestinal tracts can be empty, so it can make visceral mass a very good measure. And in addition to complete our condition measures at the individual level, we looked at the approximate composition of muscle, liver, and gonad tissues. And this approximate composition analysis determines the fraction, and that's water, lipid, protein, ash. And we certainly have data at all these levels. I'm just going to, from these fractions, we can then calculate the energy density, and I'm going to discuss this in energy density terms. This is just a picture of the apparatus used for the lipid extraction, and it's pretty obvious to do this kind of analysis. You need a lot of plugs. So just to think about these experimental measures that we looked at, going from simple to complex, we had simple measures of length, weight, from which we could calculate a relative condition factor. We also could estimate linear growth rates at monthly intervals and specific growth rates at monthly intervals. We had our bioelectrical impedance phase angle measure in there. We have the sex and maturity, and at the terminal time periods we had these additional liver mass, viscera mass, gonad mass, which we could collect, which we could calculate the HSI and VSI and GSI from. We also then had the liver total energy and muscle energy density, and the gonad energy. And just to put those measures in a time scale, what we have here is, these are the measures measured at the end of the experiment, and then at earlier months, T-1, T-2, T-3, we have these simple measures. We have this bioelectrical impedance phase analysis all along. And our hope is, as you can see, there's this big void of information here, and that's what we would love to have at earlier time steps, but these all require sacrificing the fish. And our hope is that this BIA would somehow inform other aspects of what's going on inside the fish. So we use this total gonad energy as our response metric for reproductive output. These fish were sacrificed before they actually hydrated eggs, so we couldn't estimate batch fecundity, but we thought this could be a useful metric, just at least to indicate how much energy they've allocated to the gonad at that time. And we then want to look, how can we predict this gonad energy from the whole suite of simple and the whole suite of measures we took, and then also subsets of simple and intermediate measures, and only simple measures at that time versus simple and intermediate measures at prior times, and to see how can these measures inform our predictive ability to estimate reproductive output. So for the analysis, we used Akiyuki's information criteria. We used the second order AICC to determine best models of total gonad energy from both simple measures at time t and simple and intermediate measures, and then all the whole suite, including complex measures at time t, And then we also did, again, those simple and intermediate at prior times. Once we had these best models, we also used AICC to compare them and indicate how much information is gained by including intermediate and or complex measures. So just a little results. The fish definitely, they survived this monthly anesthetization and BIA measurement. This may seem sort of trivial, but it's a new technology and it's purported to be so non-lethal, so it was. The fish grew in both treatments, and we did get the higher ration fish were tended to be in better condition. All fish were mature and developing at the end of the experiment, and just a picture of a high ration individual and a low ration individual, so we got a quite range in size. And this is just a little bit of a snapshot of what we saw in just to demonstrate that we got a range in size and weight and condition. We weren't really interested in, per se, tank effects. I've plotted them this way in this which I say ration effects. I plotted it this way just for ease of clarity, but we're more interested in the range and condition at a given size, so the variability up and down along these lines. And we saw patterns, but also a lot of variability in the phase angle, and we saw declining growth with higher growth in the higher ration fish. But since we're really interested in this at the individual level, that's where we went. When we looked at the energy density, the tissue proximate composition, we found very good, strong relationships with percent dry weight, which is fairly common. But this was encouraging that we can now use these relationships moving forward to estimate energy density simply from the percent dry mass of the tissues. But however, this BIA phase angle did not appear to be strongly correlated to any of the energy density of these three components of the fish. So this was somewhat disappointing. We hoped that it would at least indicate the muscle energy density, since that's where the electrodes were placed in the dorsal musculature. So when we get to the comparison of our best models at each time, the best model here, indicated with this lowest AICC, predicted gonad energy as a function of the ration, its sex, the final condition, the final phase angle value, its total length, and GSI. And it's interesting to see that the second best model included phase angle 1 month prior. However, there was no support for this in the AICC weights. And at 2 months prior, growth rate was important in predicting the total gonad energy 2 months later. But this analysis, we've included GSI as a measure of total gonad energy, and the two are obviously related. There's an R squared of approximately 0.72. So that's really stacking the deck by including GSI information in there. So I reran the analysis without GSI just to see, in the absence of gonad size information in there, what would we see. And when we do that, we do see, in the absence of GSI information in the model, the best model is using sex and the phase angle 1 month prior to the time of gonad energy measurement. So this indicates that there was, the phase angle had some informative ability here. The second best model that's shown here was at the time of death, including the relative condition, the phase angle, its current growth rate, and liver energy. So there's some hope here with phase angle providing some information as it's showing up in the two best models. So in conclusion, we saw food amount and quality had effects on both fish condition and the subsequent gonad energy. The energy content of muscle and liver were well estimated by the percent dry weight, and that's going to be useful moving forward with this species. The GSI provides the best energy of total gonad energy at that time. However, we can't always know what prior gonad size is. In the absence of the GSI information, the phase angle, it was useful in predicting gonad energy in the following month. However, it's significant, but it's still far from predicting in my mind, the gonad energy. The somatic growth rate at 2 months prior was informative and related to gonad energy, and also, not surprisingly, the more detailed measurements closer to the experimental endpoint explain more variable in the ultimate gonad energy. And just a further conclusion, it seems these black sea bass utilize a sort of combination or hybrid approach of capital and income strategies. Their feeding level was significant at all prior time periods, which would indicate more of a capital effect. Their liver energy at the end was related to gonad energy, which could be interpreted, I guess, as capital and income. And the somatic growth rate, relative condition, phase angle at the end were also significant, which would be indicative of an income strategy. Some of the advantages of this BIA approach: it's non-lethal, it's relatively simple. It did provide information on future reproductive output. However, we need to refine that, I believe. It may serve as a useful proxy for reproductive condition for certain species with limited opportunity to obtain detailed information, destructive measures like threatened or endangered species. And it could also help for species that are not well sampled at the appropriate time, when traditional measures are usually informative in months leading up to spawning. So some future work for obviously more validation. This is a relatively new technology, and it's not quite known what exactly some of these measures are relating to. We had hoped to indicate that the phase angle would be related to the muscle energy content, but we didn't see that. If we want to relate this to gonad energy to batch fecundity, we need to demonstrate that. And if these are validated as a proxy for reproductive potential, we could explore interannual variability in wild populations and attempt to relate that to environmental factors. We have been doing this more at the population level with several other species of flatfish, and we've seen similar patterns that I demonstrated for the tilefish in the beginning, where we see seasonal patterns in phase angle that relates to relative condition. So there seems to be something here, but before we go into a broad scale collecting this data, it would be good to further validate it. And with that, I'll just acknowledge the Advanced Sampling Technology Working Group that funded this small project experiment, and the Woods Hole Aquarium, and several people for their help. I'll take any questions.